Hi everyone, I'm the Plant Propagator and welcome to my channel. If you're interested in learning a little bit more about what I do, please subscribe to my channel. Today it is 84 degrees Fahrenheit, which is about 29 degrees Celsius. It's a warm day in Southwest Florida in early March. And my orchids are doing well, lots of good growth and new growth, and I'm really happy to see that. What I want to do today is deviate a little bit from what I normally talk about with orchids, and I want to talk to you today about science and discovery. And I want to tell you a little bit of a story about how I was so lucky to end up here. And it was science and discovery, but it was also it was naivete and making mistakes, but also being strong enough to own up to those mistakes. So you know, a lot of stuff in the sciences, a lot of stuff is good hard sciences where you start out at a beginning point and get to an end point and do all good science in between. But the reality of it is that a lot of the science that is done, it's steps. It's you make major progress and you don't have anything happen a while and you make other major progress. And there's different ways to move forward in science and in sciences. And I want to tell you a little bit of a story about some science that I did and how I ended up, again, being so lucky to be here living in Southwest Florida. For my career, I worked on gene transfer and gene expression in plants, and it was in crop plants. And there's a specific way that you perform, well, there's a few different ways that you perform gene introduction in plants. One of those involves the use of a bacterium called agrobacterium. And in order for agrobacterium to infect the tissue, there has to be access of the bacterium to wounded tissue or to part of the wall components of the target tissue. Agrobacterium is known as the natural genetic engineer. It has the ability to introduce part of its DNA into plant cells, and I don't want to go into too much detail on that unless you want to hear about that. I taught this stuff, but I don't really want to go into too much detail on how agrobacterium works. But the bottom line is, in order to introduce DNA into plants using agrobacterium, you need close proximity of the bacterium to the targeting cell. And what the bacterium does, again, is transfers part of its DNA into the cell that it targets. For years, there have been nice improvements in the efficiency. I'll get to orchids in a little bit. There's been a large improvement to the efficiency of gene introduction using agrobacterium. And I was involved in a lot of those research efforts. And at one point in my career, I was using a nice tool that I had access to. It was essentially a silica wafer that had everything etched away to leave very sharp pyramids on the silica wafer, very small, sharp pyramids. And we had a name for this, and it was called the microscopic bed of nails, because that's what it was. And the approach with that was to take this silica chip, which was the microscopic bed of nails, and touch that to the tissue so that we could wound the tissue. And so we could either get DNA directly in, which didn't work, or we could get agrobacterium, which is the natural genetic engineer that transfers its DNA to the plant cells. We could get agrobacterium to go into those large number of small wounds that we made in the target tissue. And we tried this, and we got nothing. We got nothing. It was a real disappointing thing because these were beautiful silicon wafers that were made, chips that were made, and we thought it would work. We got nothing. So we did a few experiments to try to figure out what was going on. And then what happened is we had a new Sonicator in the laboratory that we're using for cleaning dishes. And I noticed that when you put things in the Sonicator, there appeared to be some kind of jiggling going on. So we tried. So I talked to the postdoc in my laboratory on the project, and he tried to use the microscopic bed of nails with the bacterium and in putting all of this in a Sonicator. And what happened is it was miraculous. Everything was working incredibly well. So the DNA was being transferred, we had gene expression, and it was working at just a really incredible efficiency. It was working at too high an efficiency. And the reality of this is that when we looked at our tissue, the agrobacterium was actually having its impact in areas outside of where we were wounding it, the target tissue, with a microscopic bed of nails. I'll get to the point in just a little bit. It ended up that the sonication itself, through a series of good experiments and trying a number of different things, that the sonication itself was causing wounds, really small microscopic wounds all over the tissue, and allowing the bacterium to get into all these small wounds and cracks and crevices in the tissue to get very high levels of gene introduction. At that time, I was with a university. We patented the approach, and we licensed the technology to some large companies. And what that did, but it was good science behind it. We used a number of different tissue. We figured out what it was that was doing it, number one. We used a bunch of different target tissues. We did things. But the reality of it is that it was actually naive and a little bit of a mistake to try sonication with the bed of nails. It ended up not being the bed of nails that was doing anything but the sonication. And we've then backtracked to figure out why. When we published on this work, we said, well, it was a good idea. We knew that sonication would do this, and it was more of a forward-thinking approach. But the reality of it is, and I'll admit to this, is that it was naive and it was a mistake. It shouldn't have worked, and it was kind of naive to try it. But I've always done things like this. I've always moved forward and tried things that in hindsight may not have been making very much sense, and that did work. But the interesting thing and how this allowed me to end up here is that the university licensed the technologies to companies, and then the university paid me then royalties, which was good amount. So the university made millions of dollars. I made a little bit, enough to help in the purchase of my home here in Southwest Florida. And that's really one of the advantages of being on the faculty with the university at the time, is that the patent royalties do come back to the inventors. My co-inventor also did quite well. I did well. The companies that licensed the technology also did well. It allowed them to really proceed and move things forward. What does this have to do with orchids? Well, throughout my career, I've always had lots of ideas. Ideas just, they keep on coming. And even though I've retired from the university, I still get ideas and I still do things. And some of the ideas stand on their own. Some of the ideas are just based on making mistakes and being naive. And so that's kind of the balance of what you need in order to move science forward. You need to, if you make a mistake and if something works really well, you need to understand why that happened, and then you can move forward with that approach. The idea of discovery, of using these types of tools and techniques and approaches, and then understanding what they're doing is really part of the whole overall process. You need to be observant. If you make a mistake, you need to see what's going on. You need to see that, number one, things are working really well, or things are not working really well, and then you need to unravel what contributed to things working well or not well. So there's a lot of observational skills that are involved in the science that you do in order to move things forward. Okay, now, after all that, let's talk about the orchids a little bit, because I was naive and made a mistake, but I may have stumbled across something big when I shot my last video. And what I did in my last video was I showed fertilization of a Cattleya orchid. And what I did in that video is I removed the pollinia from one orchid, and I used that pollinia to fertilize another orchid. All right, and I shot the video, and what you saw, if you observe that video, what you saw was me putting the pollinia on the flower. But that was an edited video, because what happened is that was the second take. What happened on the first take, and I'll show it for you now, is that when I put the pollinia in the flower, where it was supposed to go right below the anther cap, when I put it on the stigmatic surface, part of the lip of the orchid was in the way and you couldn't see it. And I watched the video and I said, ugh, I got to do this over again. So I did it over again, and in that second retake, what I did is I needed pollinia in order to shoot the video and have it represent correctly. I needed pollinia, or I needed something the right color, the same shape and color and size of the pollinia. So what I did is I took a Phalaenopsis pollinia, and then I used that pollinium in order to fertilize the Cattleya flower for the second time. And the second time that I did it, if you notice real carefully, the pollinia aren't the same. They don't look the same with my first take and my second take. So I used a Phalaenopsis pollinia in order to pollinate the same Cattleya flower. Okay, and then you think about that, and what did I do? Now, I wanted something that I didn't want to use two different Cattleya pollinia, because that would just give me a mixed population. But what I thought I'd do is use a Phalaenopsis, because I initially thought, okay, this isn't going to work. But the reality of it is, well, I don't know what the reality of it is, but it may work. And the thing is, why can't you normally do this crosses? And there's some pollen incompatibility issues. But I'm thinking, if the Cattleya was already primed with a Cattleya pollinia, and then I followed up with a pollinia from a fowl, there may be something interesting going on. If I generate a bunch of seeds, and then those seedlings, and some of them look like they might be fowls and some of them look like Cattleyas, then I can separate those. So I made a little bit of a mistake. I needed something that I could use to pollinate the flower with that I didn't originally think would be compatible, but it might be. I followed up today and asked one of my breeding colleagues if they'd ever heard of doing anything like this, and I was told that what had happened was that somebody actually had used a Phalaenopsis, and had used the Phalaenopsis to pollinate another Phalaenopsis of the pollinia, and then removed the pollinia after day one. So maybe there might have been some pollen germination going on. Remove the pollinia from that fertilized fowl, and then use that pollinium to fertilize a different plant that it was not normally compatible with. So it wasn't quite the same thing, but it was along similar lines. So there's all these kinds of really interesting things that you can try, whereas normally you're not going to have compatibility between different plants. So that was a little bit of a long and hopefully not too much confusing video. I'll let you know what that happens. I'm excited. You know, it was just something fun to try. It was new, and it was one of those things, well, I didn't think it was going to do anything, and now I'm thinking, well, it might work, and I'm not sure anymore, but I wanted to share it with you. I've got plenty of ideas. There'll be more ideas coming on how to do some things that are new, perhaps, to the orchid world. So that's all I have for today. I hope you enjoyed what I had to share with you about naivete and making mistakes and scientific discovery. If you're interested in learning more about what I do, please subscribe to my channel, and happy propagating.